Okay, so today we're going to have the enzyme virtual lab. And so the first thing I want you to do is come to your tab within your virtual lab folder and click on enzyme virtual lab. I didn't show you how to get to the virtual lab tab. I'm assuming you understand it's under Dr. Walthall content. So this will look a bit different from the one that you did last time. So they change, they switch up, but hey, it's all the same. So the first thing you want to do is read through your instructions, which you're going to open in a Word document. And most of the time it will do this and download from the McGraw-Hill website. Once it's downloaded, it will then be a Word document. And so then we'll just open that up. And it'll tell you to open your virtual lab. The virtual lab simulation will be at the right of the screen. So on and so forth. You're going to read all background information. I'm here on 5. I skipped all of that. Answer questions 1 through 9 on the worksheet, and then follow the instructions to fill in the column to complete the virtual lab. So then we go back into our lab, and so I have plenty of tabs open here, but you want to go back to this, and you want to first do your post-lab quiz and lab report, not do it, but opening it, because it is what you'll use to fill out when you're doing the lab. So let's go ahead and open that. And here it is. So you will open up a document and it'll say virtual lab enzyme control reactants worksheet. And so you'll see, like you've seen before on your last lab, there are multiple choice questions, 10 of those. Well, 9. Usually try and do 10. Yeah, that's what I thought. So actually 14, 15 on this one. I doubled and have 14. So no, it's not 2 number 14s of the same thing. It's 14 and 15. All right. I'm human, so I change things as we go. But what we really want to look at is this here. Record your data on the number of product molecules formed per minute obtained from the virtual lab. And so this is really what you're going to be doing. So let's go back over to the lab so we can take a look and see how that goes. So if we go to the lab, we want to do laboratory exercise. And voila, this is what pulls up. So as before, all of this is your instructions here. It tells you what the purpose of the investigation is, your objectives to determine the effect of lactose concentration on the initial rate of lactase or catalyzed reaction. So lactose, of course, is the substrate. Lactase is the enzyme. Because by now you should know all enzymes end in the 3 letters ASE. So lactase is the enzyme, lactose is the substrate. If you all are lactose intolerant, it is because you have a limited amount of lactase, and so you're not able to convert lactose into its 2 sugars as quickly as others. Just as an aside to the lab. So it says you're going to determine the effect of pH on the initial rate of the enzyme lactase. So the first thing you want to do is click on the monitor to watch a video about enzymes. So we'll click here, and I encourage you to watch the video because it actually will help you.
Enzymes enable molecules called substrates to undergo a chemical change to form new substances called.
And I won't watch all of that, but you should. Again, you should. All right, so it says click on the monitor to watch a video, which we've done, and then it says click information to read about enzymes and lactose. So here you go through and you read through this. Now I know you all are tired. You're like, whatever is reading, and I see that some people don't do it until late Sunday night. That's probably a no-no if you're realizing that it takes a good minute. So you may want to take your time throughout the week and try and do this piece by piece since you have all week to conduct it, not like in the face-to-face where you're doing it that night. All right, so now it says to conduct the experiment, adjust the pH levels of the test tube by clicking the up and down arrows, and then add lactose to each of the test tubes that already contain a lactase solution. So that means that we have pH solution, and all of these are at pH 7, but we'll change this and we'll go to a pH 5 I think the lowest 3. Okay. So we'll do 5, 7, 9, and we'll do pH 11. So you're like, well, Dr. Walthall, why did you choose to do those pH changes like that? Because if I remember, my chart has pH 3, 5, 7, 9, and 11. So I want to test all those pHs. And what I'm going to do is that I'm going to test each gram. So I'm going to leave the pH 3, 5, 7, 9, 11. And I'm going to put the same amount of grams in each tube. Or you could do it differently where you do everyone at pH 3 and then you use all your different types of grams. But this is the way that I want to conduct it. And I believe it'll allow me to do that since I am the scientist and I want to do it this way. So let's see. If I take 0.5 and put it in 1, then 0.5 and put it in 5, 3, 9. You have here all this noise, right? And then I'm going to put it in 11. And that didn't work. So you have to drag and you actually have to see the test tube light up and then release. All right. So what I've done now is at pH 3, 5, 7, 9, and 11, I've added 0.5 gram of lactose. So that means that I have lactase, the enzyme, already in here, but it's at a different pH. And if you remember, there are 3 things that affect enzymes, right? It's pH, concentration, and temperature. And so for this one, we're looking at pH. And then I click on my computer and it shows me the reaction. So number 4 tells you to click the computer, gives you the reaction. Then it says click the data table. You don't have to do that because your data table is in your Word document. But you do have to put in all of these. So test tube number 1 was pH 3. It gave me a 19. Test tube number 2, pH 5, was 39. So if I go back, I fill in and say 19 for pH 3, 39 for pH 5, and so on and so forth. So many tabs open, I'm sorry. 72, 45, and 24. And then if I remember them right, 72, 40, 45, and 24. And so based upon this, you should see what is an optimal pH, meaning what is the number of molecules that form per minute. So we see I got the most molecules at pH 7, and I got the least molecules at pH 3. And if you remember anything about pH from Science 100, you should know that pH 3 is very acidic, whereas pH 11 is very basic. Hopefully you remember those things. If not, I just told you. And so you see in a very acidic environment, the enzyme lactase does not work as well. So it doesn't convert lactose as well. And at a pH of 24, it's still not working as well. The best, based upon this, with 0.5 grams of lactose, you see the best results at pH 7, giving us 72 molecules per minute. And so you'll go through and you'll do this for each of the different grams. I'm sorry, I had a brain freeze. And once you've done that, then you want to complete the questions. I would do the chart first before anything. Then go back up and do all of the questions. And then lastly, on the last page here, you want to do your journal questions. And it won't be written on your sheet, but you should do just like I'm doing now so that I know this is where you decided to answer your journal questions. And then you should do just like we've done before, and we'll go back and we'll see what our first journal question is. So some of these you have to write in yourself. So we'll go down. Well, once you're done, you want to go ahead and reset. So then you clear the board so you can start next time for your 1 gram. But we want to take a look at our journal questions. And the first journal question tells us, describe the relationship between substrate concentration, so on and so forth. So what I normally do is just take it, highlight it, copy it, and then you place it here, and you want to answer that. The journal questions must be answered. If you have not figured out yet, they are a big part of your grade. So if they are not answered or you choose to leave them out, your grade will reflect that you did not answer the journal questions. Okay, well, once you've done this, you then want to save the document somewhere, I hope in a Science 100 folder. Oh no, Biology 101 folder. Sorry. Have two of you all going at the same time. So you want to save this in a Biology 101 folder, hopefully on your computer, and then upload that document into Blackboard. I must remind you that all the time you have to upload a Word document into the lab Dropbox. You can't input data. Why? Because it doesn't give me the charts or graphs. So if you just copy and paste this thing and put it in, it's going to look messed up. I'm not going to be able to understand it, and therefore your grade is going to reflect it. Okay? Any questions, please give me an email, or you can call the office and leave a voicemail, and I'll get it via email. Remember, we are virtual this summer, so it's hardly ever that I'm in the office itself. But if you do leave a voicemail or an email, I do try and get back to you as soon as I can. All right, thanks.